Good morning, good afternoon, or good evening to all of you, wherever you are joining us from around the world. Welcome to this month's Journal of Medical Microbiology seminar series. We are delighted to have you with us today. My name is Jose Puente, Senior Editor of the Pathogenesis, Virulence, and Host Response section of JMM. I'm joined by my co-host, Dr. Kalei Mati, JMM Profiles Editor, and Hyung-Hab Watson, Editorial Coordinator at the Microbiology Society. The Journal of Medical Microbiology is part of the Microbiology Society's publishing portfolio and provides broad, in-depth coverage of medical, dental, and veterinary microbiology, as well as infection biology. As a not-for-profit society publisher, our mission goes beyond simply disseminating research. Every article we publish directly supports the microbiology community. Revenue generated through publishing enables the society to fund scientific meetings, training opportunities, grants, and professional development initiatives for microbiologists at all career stages. So when you choose to submit your work to one of the society's journals, you are not just publishing your research, you are investing in your scientific community. Before we begin, just a few quick notes. Live transcription is available for accessibility. You will prefer to turn off the captions, click on live transcript, and select hide subtitles. Today's session is being recorded. A link to the recording will be shared with all participants after the session and will remain available for 7 days. If you have specific questions for our speaker, please use the dedicated Q&A function so we can address them during the discussion segment. The chat is open. Feel free to introduce yourself and connect with colleagues joining us from around the world. And finally, we truly value your feedback. Please take a moment to complete this short poll at the end of the session. Your input helps us shape future seminars and supports our speakers. With that, I'm very pleased to hand over to my co-host, Dr. Kalei Mati, who will introduce our speaker. Dr. Mati, please. Your mic is off, Professor Mati.
Thank you, Professor Jose Luis Puente, and it's also a pleasure to see our editor-in-chief, Matthew Diggle, here. Thank you for joining us, Dr. Diggle. Friends, researchers, ladies and gentlemen, it's my genuine pleasure to introduce our next speaker, Professor Peter Dedon of MIT. I first encountered, and this is Pete doesn't know this, Professor Dedon doesn't know this, the way many of us discover our most interesting colleagues buried in a list. I had compiled a roster of scientists connected to Pseudomonas research 2017, and there was Peter Dedon, and I look at the email address, and it is MIT address. So an MIT engineer who had apparently wandered into the microbial world with the same cheerful purposefulness with which he seems to have navigated his entire career. That alone made me curious, but curiosity becomes something more than I had the fortune of meeting Professor Dedon in person at the 18th Asian Conference on Transcription in Bangkok this past December. What struck me immediately was not just the depth of his science, but his disarming honesty about how we got there. Here is a man who hated chemistry in high school and majored in it anyways. I think when we hate something, we put extra hard work because we want to do well. Who earned his PhD in pharmacology, largely because his advisor changed universities, and he simply followed. Who will tell you was that with a straight face that his research program in epigenetics and epitranscriptomics was the product of a grand design, but not of a grand design, but of bumping into the right students and scientists at the right moments. And that's very telling. By his own admission, his life in science is less a carefully charted trajectory and more, and I quote, a very enthusiastic accident. And I find that profoundly reassuring, and I suspect you will too. Because behind the self-deprecating candor is a scientist who spent 3 decades doing genuinely original work, Professor Dedon is the Underwood Prescott Professor in the Department of Biological Engineering at MIT. He leads the Antimicrobial Drug Resistance Program at the Singapore MIT Alliance for Research and Technology. And he's a founding member of the Chulabon Graduate Institute here Institute in Bangkok, which made his presence at the Asian conference all the more fitting. What Professor Dedon Group has built over these decades of productive accidents are platforms, rigorous, elegant, analytical, and informatic platforms for asking questions that most of us hadn't thought to ask. And, it's also not they haven't thought to ask, it's hard to answer in my mind. How one nucleic acids modified, when, under what stress, and what does that do to the cell's ability to respond, adapt, and survive? His work sits at the intersection of chemistry, biology, and engineering in the space of central dogma left largely unexplored, not what is expressed, but when, how much, and in response to what. That turns out to be a very large and very important space, especially when the organism in question is a pathogenic microbe or human try to avoid antibiotics. Professor Dedon's work in epigenomes and epitranscriptomes has revealed that microbial cells are far more dynamic, far more cells in general, far more sophisticated in their regulatory responses than the central dogma, elegant, simplicity might suggest. He has built the tools to see the dynamism in molecular detail. He is, in the truest sense, a chemical biologist, one who brings the precision of analytical chemistry to bear on the messiness of living systems. And he does it with collaborative spirit and intellectual generosity and with genuine delight in discovery that I experienced firsthand in Bangkok. I am very glad, on a personal note, that he was in the list, then a conference, and this occasion brought Professor Dedon into our orbit. Please join me in welcoming Professor Dedon today.
Thank you, Kalai. Very nice introduction, and it is true. A lot of what I want to talk about today is an accident, and I'll point out where the accidents happened and why that's important. So I will start sharing my screen here and start the slides. Hopefully you can all see that. Excellent. Okay. So, yeah, this is great. This is really fun. It's a big honor to be talking to such a big international group. Zoom is such a powerful thing now for bringing the world together, and this is just a great opportunity. I want to talk about the work that's been done for over the last about 15 years now. The current groups that I have at MIT, and as Kalai mentioned in the SMART program, we are one lab. We just happen to be 12 time zones different. I'll be going to Singapore in a week, as I do 4 times a year for about 20% of my effort. The things I'm going to talk about today are split evenly between the two groups in terms of the brilliance of the students and the scientists. So, as Kalai mentioned, the whole concept of underlying our work is nucleic acid chemistry and biology. I'm a MD. I don't practice. I have a PhD in pharmacology. All that means is that I really do basic science and try to translate it, no pun intended, into practical solutions. So the idea of the central dogma, 20,000 genes in a human, 5,000 in a bacterium, they get turned into messenger RNAs and proteins. But the things that actually schedule that are the epigenomes and the epitranscriptomes that I want to focus on today. And I'm going to move fast because I couldn't figure out what to talk about. I have so much microbial work in both epigenetics and epitranscriptomics, so I'll try to cover as much as possible in a moderate amount of time here. So we do a lot of work in the DNA damage realm that I'm not going to talk about. We have discovered over 100 new kinds of DNA damage that happens inside microorganisms, in humans, in different tissues. We've got a wonderful group of colleagues doing sort of systems level DNA damage analysis. I want to spend a minute talking about epigenetics. In humans, this is a few, 4 or 5 DNA modifications, and then 20 or 30 histone protein modifications, all of which regulate transcription. When do we transcribe? How much do we transcribe to get all those messenger RNAs and the non-coding RNAs that are needed? So I want to talk about the epigenome, especially microbial epigenomes, with a great team of people. Now, this is a fun story that goes back 60, 70 years now. A German chemist by the name of Fritz Eckstein invented phosphorothioate DNA modifications where a sulfur replaces an oxygen in the phosphate sugar backbone of the DNA. So he invented this back in the late 60s as a synthetic modification of oligonucleotides. Well, you fast forward now where all of the FDA approved oligonucleotide therapeutics all have phosphorothioates in them, in their backbones. The reason is that it turns out that these phosphorothioates resist nuclease degradation. So by putting them into the backbone of the oligo, they protect them against degradation in the blood or in the spinal fluid, wherever they're being injected, and they prolong the pharmacokinetics of these compounds. So Fritz has made a miraculous discovery here in inventing this. He didn't know at the time that that was going to be the case. Now, you fast forward 40 years, what we did was to discover that phosphorothioates are natural products. They're made in bacteria and archaea. Humans don't have them. Higher or eukaryotes don't have them. And then here's the accident. Wang Lianrong was a student at that time at Shanghai Jiaotong University, walked into my lab one day and asked if she could come and work for me. She knew that sulfur was getting into DNA, but they didn't know how, and they didn't know the chemistry. And I said, well, I don't have any money to support you. I'm sorry. It sounds really fun. She said, oh, I don't need any money. My husband's a postdoc over at Harvard. I'll work for free. And it's sort of one of those moments every professor says, well, okay, why not? Let's do it. And sure enough, through a series of analytical techniques that we had in the lab, she mastered them quickly, and we got a very nice paper showing that the DND gene family, which I'll talk about in a moment, synthesizes phosphorothioates in bacteria. It was ironic then that I met Fritz Eckstein for the first time. He wrote the News and Views story that accompanied this piece, making the joke that nature beat him by 1 million years to inventing phosphorothioates. Well, the nice story for him is I went to his 90th birthday party in Göttingen, Germany, back in 2022. I was the only biologist, biochemist there. The rest were industry people. And if there's anybody that's on a short list for the Nobel Prize, it's got to be Fritz because of the impact of his work on modern therapeutics. All right, so phosphorothioate modifications are part of restriction modification systems that everybody knows well in microbes. They're a complicated system with 5 enzymes that cut the strand, activate the phosphate, put in the sulfur, religate the strand. And then there's a cluster of genes that do the restriction. Now, what's unusual is that only 10% of the consensus sequences in a bacterium genome are modified. So how does the restriction enzyme avoid cutting at unmodified sites? We've got a model out that says it's a density-dependent sensor system. We don't know how that works. But if you drop the modifications below 10%, you start to get nicking in the DNA. So there's some sort of a way that the restriction enzymes are keeping track of how many you have and where they're located. And that is part of the process. Now, zooming forward now, past the basic science, we discovered starting with mice and then moving into humans that about 5 to 10% of the bacteria in your gut microbiome have phosphorothioates in them. And we use tools now like mass spectrometry. Remember, the phosphorothioates make the DNA resistant to nucleases. So if we digest genomic DNA with a nuclease, we're left with dinucleotide limit digest material. So we take the bacterial genome, digest it down. Each of the spikes on this HPLC chromatogram is a different dinucleotide. We have 16 synthetic standards of the 16 possible combinations of 4 nucleotides. And we use those then to digest a bacterial genome and identify which dinucleotides does that bacterium have. That reflects the consensus sequence. Consensus sequence might be 4 or 6 nucleotides. We get the 2 surrounding it. We've also developed a variety of sequencing technologies now to map the modifications in the genomes. And here's our first effort with Pact Bioscience direct sequencing, showing the presence of a phosphorothioate in a genome along with an M6A, another epigenetic mark in the bacteria. So we use these tools now to go characterize how many bacteria have phosphothioates and which bacteria are they. And what we found then in the gut is that about 10% of the bacteria have this. And here's an example now with 11 healthy human donors. Everybody from the lab volunteered. We won't tell you who they are. When we take the fecal DNA out and digest it, everybody has a different signature of phosphothioate dinucleotides, different combinations and permutations, different relative quantities, but it's a signature that reflects the uniqueness and signature microbiome that we all have. If we look over time, and this is over about a period of about 9 months, you see these fluctuations in the levels of the phosphothioates. This is a person that has 3 dinucleotides, reflecting the 10% of the bacteria in their gut that have these modifications. This variance is just due to the daily changes that happen in diet and things like that. No big significance here. It's Taylor's law, as the microbiome people have quantified this mathematically. Now, we use our sequencing technology then to say, well, which bacteria in your gut have these phosphothioates? Normally, in an adult gut microbiome, about 25% of the bacteria are Bacteroidetes phylum. Firmicutes are the big ones at about 50%. However, when we look at phosphothioates, far and away the biggest group of bacteria possessing phosphothioates are the Bacteroidetes. Firmicutes now are coming in at a very distant second. So we have a big enrichment of these phosphothioate bacteria in the Bacteroides species. Now, here comes the medical part where it gets translational. I started my career in inflammation chemistry, looking at DNA damage. We know that when an infection happens, macrophages get activated, neutrophils get activated, and they make a variety of reactive oxygen, nitrogen, halogen species to kill the bacteria, phagocytosis, and then dump a bunch of toxic chemicals on the bugs to kill them. Well, those chemicals are dispersed all throughout the tissue. They cause DNA damage and mutations in humans. We thought that all these phosphothioate-containing bacteria, because they have sulfur, are going to get killed by all these reactive species. Sulfur is very easily oxidized. Sulfur is a potent nucleophile, much more than oxygen. And so why you would put sulfur in a genome is beyond me, because it renders the genome susceptible to damage by all these toxic chemicals. So I partnered with a gastroenterologist at Brigham and Women's Hospital, Josh Korzenik, who's the head of the Crohn's and Colitis Center. And I pitched this idea to him. I said, I'll bet that in an inflammatory bowel disease with tons of inflammation, that all this phosphothioate bacteria are going to get killed off. And maybe they're important for protecting against that. And he said, no, hang on a second. You're forgetting something. When people have inflammation of the gut, the mucosa sloughs off into the lumen. The mucosa in your gut and in all the mucous surfaces is highly sulfated, lots of sulfated glycans. So he said that the bacteria may be swimming in a sea of sulfur metabolites. And he said your phosphothioate bacteria may flourish and grow significantly with all of that sulfur metabolites. So we got a bunch of samples together. We put a bet on this, and he won the bet. As it turns out, you can see the controls over here have phosphothioate levels measured by mass spec on the order of about 10 per million nucleotides. People with Crohn's disease have upwards of 200-fold increased concentration of phosphothioate-containing bacteria in their guts. And that's whether it's active Crohn's disease or inactive. We don't fully appreciate why some people don't have high levels. And we're investigating that now, expanding this group. But there's clearly a subset of these Crohn's disease people that have huge enrichment for phosphothioate bacteria. This satisfies one of the first of the 3 principles of inflammatory bowel disease, typically you have 1,000 different microbes in your gut. People with IBD have greatly reduced diversity, maybe 200 or 100 left. So all of a sudden, our enrichment may be part of that reduced diversity. The second thing is that Josh is finding that in the reduced diversity, we're enriching for bacteria that make hydrogen sulfide. Hydrogen sulfide is toxic to the epithelia in the gut and kills them. The third thing is that we reduce the number of bacteria that make butyrate, butyric acid, short-chain fatty acid, that interestingly, human epithelial cells in the gut lining use that for energy more than they use fatty acids coming from the blood. So all of a sudden, you're hit with hydrogen sulfide, you're injured, and now you don't have the energy to repair the injury. You die, and you have inflammation in the gut and the horrors of inflammatory bowel disease. So we went back and did we do a lot of informatics and genetics and genomics as well as measuring molecules because of the systems level things here going on. And what we found in a nearest neighbor analysis was that phosphorothioate bacteria have very significant enrichment for hydrogen sulfide producing genes. So I think we're seeing that phosphorothioate bacteria are part of the hydrogen sulfide producers that get enriched in the inflammatory bowel disease, and this may actually be causative or contributing to the symptoms of inflammation. So now, as a drug designer, I'm looking at this and saying, I'm going to start making molecules that kill phosphorothioate-containing bacteria selectively, leave the rest of the microbiome alone, and see if that relieves the symptoms of inflammatory bowel disease. So this is really a lot of fun from this point of discovering these things, now being able to potentially come up with a treatment. I'll point out that the bacterial microbial genomes are loaded with DNA modifications. Cui Liang is the head of our mass spec facility and a former postdoc of a colleague of mine at MIT. He's now discovered 10 different DNA modifications in bacteria and phage where you have a 7-deazoguanine. And all different permutations of that. And in fact, some bacteriophage, 100% of their guanines are replaced with a 7-deazoguanine. So it's no longer even DNA anymore. It's deazoguanine DNA, not cut by restriction enzymes then. Well, this deazoguanine motif is also an RNA modification found in almost all organisms, cuisine. We'll talk about in a minute at the wobble position of the tRNA. So there's crosstalk between RNA epigenomes and RNA epitranscriptomes and the DNA epigenomes. So interesting, fun part of the epigenome world here. Okay, so let's switch now to talking about the epitranscriptome. Here now we're talking about a world of all RNAs in all organisms are proven or predicted to have modifications in them. Humans have 250,000 different transcripts. Microbes have 10 times less, 25,000 different transcripts, all of which are modified, whether they're coding transcripts or non-coding transcripts. And in this world, it's more than 170 chemical structures now known to be modifying the RNA. And these are enzymological insertions, very intentional and very functional. And I love, as a chemist, I just love a diagram where there's 100 chemical structures. It's kind of a woohoo moment here of cool structures. That's growing. The list is growing about 2 or 3 per year. I think Tom Suzuki's going to introduce another 5 soon. Everybody's waiting for the trumpets to sound. tRNA is the most heavily modified form of RNA. This is about 10 to 15% of the nucleotides are modified. Ribosomal RNAs are next in line at about 2%. Messenger RNAs are really popular, the messenger RNA modifications, just because they were some of the first to be studied intensively. They're very low density. There really aren't that many in them. Now, all organisms possess somewhere between 30 and 60 modifications. Microbes, a few less. Humans, right now, it's about 55. Give me a second here. There we go. I like to focus on the transfer RNAs because they're the most heavily modified, and they are part then of reading the codons in the messenger RNA and part of translation very directly. Bacteria, at most 200 tRNA genes expressing maybe 30 to 50 tRNA isoacceptors each reading a different codon and carrying the amino acid on them. Humans are more complicated, have 500 genes expressing 250 tRNA types, and these are then isodecoders, where they have the same anticodon but a different RNA sequence in the body. All the microbes have 8 to 10 modifications per tRNA, and the highlighted orange dots here are where they're located in a generic transfer RNA structure. You can see that the anticodon there in the anticodon loop, the most diversely modified position is the wobble position. And it just makes intuitive sense. Wobbling is the reading of different codons by the same tRNA. If you put a modification on there, you can switch the wobble. You can force it to read one codon, or you can make it wobble and read multiple so that you get significant diversity. There's a huge information content in the modifications in the tRNAs. If you give me yeast tRNA from an exposed cell culture and an unexposed, I can tell you with about 85% certainty and specificity what the chemical was that you were exposed to. We did a machine learning model on this years ago. And the information in there, and I'm going to show you this in a minute, is part of a stress response pathway. Dysfunction of tRNA modifying enzymes is linked to over 100 human diseases. And I want to show you this coming up. Because bacterial enzymes are very different structurally than humans, and they have modifications that humans don't, all of a sudden you've got juicy antimicrobial targets that are very specific to the bacteria and don't occur in humans. So what I want to talk about here is a systems level behavior that I have been working on now with Tom Bagley for the last probably 20 years now. Here's another one accidental moment, and this is fun. Tom Bagley was a postdoc at MIT. He moved to the University of Albany to take a faculty position and had a transitional grant. He had to have an advisory board for that grant. He invited me to be on there because he knew I did mass spec stuff. And I showed up to his first presentation for his grant. And he told this story of how yeast, when they get exposed to alkylating agents, one modification increases significantly in one tRNA, and somehow that causes translation of messenger RNAs needed to respond to alkylating agents. And I thought, that is cool. Really, he was going to have a good trajectory for his career. And I asked him, I said, aren't there like dozens of those modifications in the RNA? And I said, I can measure those on the mass spec all at once, probably. And so I did what you're not supposed to do as a mentor. I resigned from his advisory committee and became his collaborator. And we now have been collaborating 50 papers, more than 50 papers, about 10 or 15 grants that we've had together, collaborating for 20 years. So it's the most wonderful resignation I've ever had at being a mentor. So what we've done here now is to do multi-omic analysis, measuring all the RNAs, ribonucleomics, measuring all the modifications. Tom is big on genomics and quantifying codon usage patterns, and we'll see how this works, and proteomes to measure changes in translation. What we have here then is when a cell gets exposed to a stress, the tRNA pool, both the tRNAs and their modifications, reprogram. We change the number of copies of the tRNAs. The modifications reprogram, as I told you earlier, they change in response specifically to the stress. The reprogram pool then goes and reads specific codons better or worse. And what we get then is the pool selectively translates messenger RNAs coming from gene families that give you the stress response proteins you need for that specific stress. So it's a way for the cell to regulate its phenotype without waiting for big transcriptional upregulation. We can pick messenger RNAs that have the right synonymous codons, and we can translate them through the reprogram pool and get the proteins we need to survive. DNA repair, cell cycle control, all of that unique to each stress. So let me walk you through this then with some microbes. What I want to tell now is how this works with mycobacteria exposed to hypoxia. This is what happens when a Mycobacterium tuberculosis, you breathe it in, it gets eaten by a macrophage, the granuloma forms around that, no blood supply. So you don't have any oxygen, you don't have any food. The bugs go into the classic dormancy where they stay non-replicative for 20, 30 years. They can then come out again, have a second infection that is deadlier than the first. So this is the story of sort of mycobacterial dormancy. Well, the first student that I had in Singapore worked on this, and nobody had ever studied modifications in mycobacteria. So he had to go through and characterize all of them. He's got 40 here that he studied. There's more than that. There's about 45. And so he then set up a mass spec method to quantify these. And you're seeing the peaks now of individual ribonucleosides. When I take the RNA out, hydrolyze it down to nucleosides, squirt it on the HPLC, they get separated, they feed into the mass spec where we quantify them. Now, when we look at this and we start crunching the data, the columns here are days of hypoxia in a Mycobacterium bovis culture where we just screw the lid shut and they run out of oxygen. And so you can see when we have oxygen, there's certain modifications that are higher than the others and certain modifications that are lower, the green. Those are the oxygen conditions. When we first seal the bottle and then 4 days after we open the bottle again and get oxygen in there. But early in hypoxia, this switches. So the ones that were up go down, the ones that were down go up. And so these are patterns of change as the cells respond to the hypoxia. The 4 white lines are the 4 known century-old microscopic behavior patterns of mycobacteria that are under hypoxia. So we're just replicating what we already knew, but we're looking at a different system. So Jokian picked one modification, CM05U, that goes at a wobble position on one of the 3 tRNAs that have a threonine on them, and it has the anticodon UGU. And now it's a wobble, U is modified. That is going to read one of the 4 synonymous codons for threonine. Turns out that by putting CM05U on that wobble position, it locks it into reading the ACG codon. So there's part of our reprogramming. The tRNAs now get wobble modifications that are new. Okay, here's another tool we use, quantifying the tRNA members in the pool. And this is 13 of the isoacceptors in Mycobacterium bovis. There's the time course here. And you can see the absolute quantities of these tRNAs. Here's a tRNA that's abundant. Then it goes down and comes back up when there's oxygen. Well, there's some tRNAs that are very rare. Down here, the rare ones. Some go up and then come down. So we're watching the reprogramming of the pool, numbers of copies of tRNAs in the pool as a function of the hypoxia. So there's our reprogramming. Now, what about the codon biased translation part of this? Well, this is where Tom Bagley now has developed software to quantify codon usage patterns in every gene. All right, the columns in all these heat maps are the 61, each of the 61 codons that code for an amino acid. The rows are the genes. Purple means that the codons are overused in that gene set. Yellow is underused codons. And you can see humans have about half their genome are heavily codon biased. Yeast, it's about 10% of the genome. Mycobacterium bovis, again, about 10% of the genome or more is codon biased. And in mycobacteria, there's 2 groups of opposite codon bias. Well, this top group are all the genes you need to go to sleep. It's the DOS regulon that regulates early microbial response to hypoxia. Okay, very cool. Well, now what we've discovered more recently is it's more than just some enriched modifications in some genes. What we found is that gene ontology categories of gene function, each different category has a different pattern of synonymous codon usage, not one codon over or underused, but patterns of codon usage that distinguish them statistically significantly. So there we have that messenger RNAs with these patterns that match the reprogrammed pool. They get selectively translated to make the proteins we need to respond to that stress. What are those proteins to survive hypoxia? Cell cycle regulators. Some of the proteins we're going to shut down are metabolic proteins. We don't want metabolism. Ribosomes degrade and go away. The cells stop dividing and they become dormant and survive for 30 years and then come back to life again later. So how do we know that this really happens? Well, again, we can go do proteomics on the aerobic and the anaerobic bacteria, and we do multivariate statistics. This is a loadings plot, but when we did the scores plot, the blue area denotes where upregulated proteins would be segregated. So these are all the proteins that went up in hypoxia. the loadings plot shows that in those proteins, the genes for those proteins are enriched, heavily enriched with the AAA synonymous codon for lysine. In all the proteins that went down at that same time point, they're enriched with the synonymous codon AAG. So there's an alternative genetic code. If you want to turn a protein on in early hypoxia in Mycobacterium bovis, enrich with these codons, AAA, cysteine TGT. If you want to turn proteins off, put their synonymous counterparts in so that we can now regulate which proteins are going up and down and control the phenotype. Okay, now that's a lot to digest. It's a complicated system. I want to now tell you how we want to move this now to do high throughput. So all of that work done previously to take dozens and dozens of samples was all done manually. We now want to automate this because I want to go screen a 5,000 strain knockout library of Pseudomonas. So how do you do that without making grad students get retirement plans so they can work for 20 years? We don't do that. So Jingjing Sun and Junzo Wu have been largely responsible for automating all of the things I just talked about, taking a 96 well plate, processing the RNAs, purifying them, getting them ready for mass spec analysis. We now have a gel-free library preparation technique for next-gen sequencing libraries, so we can automate next-gen sequencing. We also, I'm going to tell you in a minute, have a new mass spec sequencing technology that is sort of the one ring to rule them all from Lord of the Rings here, to do the sequencing and the modification mapping. So this is Jingjing is a brilliant technologist, and what she did was, first of all, you put cells, tissue, anything you want in the 96 well plate. She came up with a way to lyse the cells very quickly so that we don't wait for things to reprogram. She can purify each type of RNA, small RNAs, ribosomal RNAs, messenger RNAs, using bead-based technology, get them very pure. She then has these TCAN liquid handling robots. For example, hydrolyzed the RNA to nucleosides. We can inject it on the mass spec to do what we saw earlier. And now she can do this 5,000 times, and we get big data plots that we'll then mine to get meaningful biologic information. Just published this last year in Nucleic Acids Research. Very nice story. Okay, so her first project was to say, what happens if we take a bacterium and start knocking out individual genes? How do they affect the pool of ribonucleotides? How does it reprogram when the cell gets stressed out by losing a gene? And so we took the 5,800 strain transposon mutagenesis library, individual strains now, not a big combined tagged library, and assayed them individually to ask, what are the 50 plus modifications doing when I lose this gene? And so we know that there's dozens of RNA modifying genes in there, so we have automatic controls to show if I knock out an RNA modifying gene, the modification should go down. So it's a beautiful story. She set the robot up to do these. It took 2 months to go through 60 plates and do all the analysis and the data analysis. Here is one of those LCMS chromatograms. She's able to get 52 modifications analyzed in less than 3 minutes now. For the paper, it was 6 minutes. She's got it down to 3 minutes. And that's important to get the throughput. The faster you can run this, the more samples we can analyze. And here's the end result of scanning 5,800 strains. Each, along the x-axis, is the number of the strain. The column is the 52 modifications. Each dot is a modification. Most of them are just gray. Gray means they didn't change by more than fourfold. We don't want to study those. Let's go for the big changes first and see what's out there. So the red dots are fourfold up, blue dots are fourfold down in terms of those modifications. Big, giant data set. We actually got a software engineer to help us set up the data to take all that and crunch it and do it in an automated way. Okay, now you apply multivariate statistics. And this is principal component analysis, heat maps, all the same stuff. Software does this for you. There were 135 genes that cause these fourfold up and down changes. The columns are the individual RNA modifications. And I like to do what I call blobology. I can do the statistics, but I think it's more fun to just go pick out the most colorful changes, right? Well, here's a big upregulation, an orange blob of modifications that went up when this gene set was knocked down. And at the same time, modifications that went down The interesting part now comes when you look, they're all I6A family modifications. That's a position 37 modification. IO6A, MS2, IO6A, MS2, I6A. Interesting. Okay, now dive into that. Let's go look at this. So here's the pathway that makes this modification. I6A is present at position 37 in the anticodon loop. There's our wobble position at 34. There's our anticodon. Position 37 has been known for decades to affect the way the tRNA binds in the ribosome. So it's important. It's an important modification. Start with an adenosine. MIA-A pops an isopentenyl group onto that. MIE oxidizes it now, so we get a hydroxyl group, I6OA. MIA-B comes along and puts a sulfur on that. So now we have MS2I6A can get oxidized to MS2IO6A. All these interesting permutations of that position 37 modification. We know that the system works when we knock out MIA-A, all the I6A modifications drop to 0. Great. If we knock out MIA-B, all the sulfated ones drop, and the other unsulfated ones increase. Great. Positive controls, the system works. Now, what we wanted to ask was, all right, let's go look at what happens to MIA-B modified ribonucleosides when we start knocking out genes. So we took a ratio of genes that caused I6A to drop by taking the ratio of MS2I6A to I6A. And we took everything that caused that ratio to drop to less than 0.5. Which genes did that? Now you go look and see what families these genes fall into, and you get a map like this, a functional map. What you see is the genes that are affecting MIA-B activity are falling into classes: purine pyrimidine metabolism, nitric oxide reductase, multi-drug efflux transporter. So maybe there's an antimicrobial resistance mechanism here. Electron transport, iron transport. What is going on? All these diverse pathways. Well, now you make it a little more meaningful by mapping out the pathways. Here's MIA-B. It takes I6A and converts it to MS2I6A. MIA-B, and here's the chemistry now. You got to put your chemistry hat on. MIA-B is an iron sulfur cluster protein. It takes iron to make them. If you're short on iron, MIA-B doesn't work well, and we get less MS2I6A, more I6A. So there's a first signal that connects to tRNA modifications. It says, I'm short on iron, change the tRNA, reprogram the pool, let's translate iron transporter genes. We're just now testing that hypothesis. All right. We need electron donors to keep that iron in the iron sulfur cluster at the right +2 valency. Well, what if you're missing these factors that are involved in electron transport? We need a vitamin B, to do sulfur transferase, to put the sulfur in. Here's an interesting one. When the bugs grow anaerobically, they're going to reduce nitric oxide to get their electron transport done. Nitric oxide is a well-known potent inhibitor of iron sulfur cluster enzymes. All these things are going to affect the activity of MIA-B. When we then have these stresses, they switch the pool. Either we get more unsulfated modifications or more sulfated modifications. We don't know yet how that switch of these position 37 modifications alters translation, but we're working on that. But the hypothesis is that it subtly regulates translation of specific messenger RNAs needed for iron sulfur cluster repair. And so there's your signal between anaerobic metabolism, for example, and a switch to proteins needed to survive that state. Okay, I'm going to try not to go too long. I want to talk about something new. Now, we're going to jump from microbes to humans. We're not being elitist snobs here and ignoring the microbial world, but there's a new program going on called the Human RNome Project. It's this next step after the Human Genome Project. It has started in 2021. There was an NIH workshop that I helped organize. That led to a National Academy of Science meeting where they pronounced, and when the National Academy of Science pronounces something, it's like it's coming from God, and you'd better believe it and take note of it. They published a paper talking about what are the tools and infrastructure we need to do what's in blue up there. The RNome project is map all 55 human modifications in all 250,000 different RNAs in all types of cells in all human diseases. And, that's the one where you go, what? It's impossible, right? Of course it is. But let's set our impossible Don Quixote mission and go do this. Let's set out to do this. But what we do then is discover what's going on with these complicated epitranscriptomes and how they're affecting disease, and then we can come up with cures. So we've had a couple of workshops now. The next one's in Toronto. You can go to the website for the Human RNome Project, and if you're interested in this kind of thing, it applies to microbes too. I do the microbial work, and we got to do the microbial RNome project if we're going to cure human disease too. Okay. And here's the deal. You saw this earlier. This is just repeat. So many modifications, so many different forms of RNA, so many ways that they shift around. In messenger RNAs, the M6A in messenger RNAs in eukaryotes moves. Well, why is it where it is? What's it doing when it's there? Nobody knows. So we got to figure this all out with the RNome project. Well, the National Academy of Science laid out what are the tools we need to do this. We need synthetic RNAs. We need ribonucleoside standards. What are the companies making them? Who's going to make them? Who aren't making them? We need infrastructure. One of the infrastructures we need are the tools to do this. Now, the tools needed to map all the modifications are three of them. One of them is nanopore sequencing, long read single molecule sequencing, where you can take one long messenger RNA and map the modifications in it so that in each molecule, copies of that molecule of RNA all could have different modification patterns in them. So we got to know that. Then we want the short read, but Oxford Nanopore technology is not specific. We can't tell which nucleoside is in there. It's not very good. It's getting better. We need short read technology like next-gen sequencing to identify the modification and do single nucleotide mapping. We then want to come up with a mass spec-based method that I'm going to talk about, because with next-gen sequencing, you need 50 different methods to map the 50 different modifications. Give me a break. I want one method to map all modifications in any RNA. That's what mass spec is all about. So here comes Junzhou Wu. And what's fun in the world, Junzhou is married to Jingjing. So this dynamic duo is doing very interesting work in moving things forward on the RNome. What Junzhou has done is to create an RNA sequencing technology that's just like proteomics. And I don't know how many of you have done proteomics, but if we want to analyze 10 samples under different conditions, we use iTrack or TMT labels. You fragment the protein, put the labels on, then you mix the samples together, squirt them in the mass spec, and you get your data back and your proteins up and down, and really powerful tool. And you can map the modifications on the proteins using proteomics. Well, Junzhou is going to do the same thing with RNA. And what he has, and then there's all kinds of challenges. First of all, this fancy chemical diagram on the left is the fragmentation pattern in the mass spec of a nucleic acid. Proteins have completely different fragmentation patterns. It's different chemistry. And so all these bars, all these lines here are fragments when you take an oligo and smash it to pieces in the mass spec. And this what happens with peptides. And the trick is to convert those signals into a sequence. You need entirely different software to do that compared to proteomics. And when you do that, you can puzzle it back together into a sequence. And then there's a modification that we can map and quantify it. Unmodified oligo, modified oligo. So all the challenges now here that RNA and DNA shatter at much lower energy than peptides. So your tags, like iTrack, fragment at high energy to give you a reporter. You can't use them for RNA and DNA. We had to have brand new ones. Junzhou invented some new ones. We need ribonucleases to cut the pieces into mass spec friendly piece sizes. Ribonucleases typically shred the DNA because they have single nucleotide specificity, most of them. So lots of challenges. And I want to show this picture here of the need for isobaric tags. This is just like proteomics. You take your sample, it's got RNA in it, you digest it to oligonucleotides, you add your tag covalently, You then have these different reporter tags, you mix them all together and squirt them in the mass spec. What happens is all the RNA molecules that are the same but coming from different samples have an isobaric tag. The tag weighs the same in all of them, so they all are mixed together and have the same M over Z value. They go get fragmented in the fragmenting chamber. The reporter molecules fall off, and I'll show you that in a minute, but the RNA itself is the same for all of them, fragments, and we get our sequence. Now we know what that oligonucleotide is. If we look down at the low molecular weight, here are the relative quantities of the RNAs in that sample. In sample 1, it was low, it goes up, and then comes down in sample 4. The reporter quantity tells us how many copies of that messenger RNA came from each sample. That's exactly what we do in proteomics to quantify and multiplex. Multiplexing saves so much money and time. Okay, so what Junzo has done now is to develop new ribonucleases. He's invented several of them that have dinucleotide specificity. He'll sell those to New England Biolabs or Sigma or somebody to sell. He's made new isobaric tags. He's got a 4-plex now that I'll show you. I won't show you. I'm going to show you iTrack, but he's not talking about these yet. He's working with Agilent Technologies to come up with new chromatography. Peptides are great on reversed phase HPLC columns. They separate very nicely. Oligos are negatively charged, really uniform physical chemical properties don't go well on reversed phase. We need helic, hydrophobic interaction, lot of chromatography. Very different kind of chromatography. Finally, you need new software, completely different software to convert signals to sequences. So he's working with OpenBiome and Sam Weinstein at OpenBiome to write new software to interpret the data. Let's walk through an example of this. We're going to take some of those Pseudomonas knockout strains. He just picked a bunch and a wild type for comparison, purify their tRNAs, fragment them with ribonuclease T1. Fine, we're going to shred most of the RNA, but we get enough usable. Put the tags on. We're going to do iTrack tags here. Mix them and do the mass spec. What he gets about 30% coverage of all the tRNAs in that pool. Not bad for your first try at something. And this confusogram just shows you each of the different oligonucleotides he could detect. The columns are how abundant that is in each of the mutant strains from the Pseudomonas. These are all tRNA modifying enzymes. There's MIA-B. There's our friends MIA-B and MIA-A from the I6A pathway. And we see that when we knock them out, some of these oligos go away. Now, I'll show you a perfect example of that. Here's an example. If we pick one isoacceptor, and that's the tRNA that holds cysteine and has the anticodon GCA. There's the anticodon down there. Oh, no, sorry, right there. There's position 37 that we talked about. He can now go through and see some really interesting things here. You see the key shows you the oligo that he's going to measure that can have either IO6A, I6A, MS2I6A, or MS2IO6A. Amazingly, the same sequence can have each one of these different 4 things in subsets, all right? All right, if we knock out MIA-A, all of those oligos go to 0. The fold change drops. You can't detect it because there's no I6A. Great. Then we knock out MIA-B. All of a sudden, IO6A goes way up and I6A goes way up. Why IO6A is more abundant than I6A, it's interesting. Okay, what's going on there? There's interesting new things to do. Now, what happens when MIA-B is knocked out? All the sulfurated ones go down almost to 0. Not quite to 0, and we don't understand that. Is there some leak through on the expression? But MS2I6A drops down and MS2IO6A drops down. And what this is saying is that in a pool, in Pseudomonas, the pool of tRNAs has this one isoacceptor where different fractions of it will have different modifications to do translation. Here's another thing he discovered by doing this mutant analysis. If I have MS2I6A present at position 37 in the same molecule, I'm probably going to have M2A at position 38. That's a newly discovered modification at that site. M2A is well known. It's never been known at position 38. He found it using the mass spec. Turns out TRIM5 needs MS2I6A in order to modify position 38 with M2A. Cool. We found a dependency, a crosstalk. Here's another one. Let's say MIA-B is inhibited and we only have I6A. TRIM-L requires I6A to put 2-O-methyl-C at the wobble position of another tRNA. Go figure. Here's a way where these modifications now are regulating what else is present in them. So there's a progressive decoration and reprogramming that goes on. Turns out you need these wobble modifications on the tRNAs that read the codons that are enriched in the MIA-B gene. Once again, if we shift the tRNA pool to have these tRNAs, we're going to translate MIA-B messenger RNA and get more MIA-B. When? I don't know. When there's some stress that needs MIA-B. So really interesting, beautiful information comes out of this kind of analysis now. Okay, I'm going to go quick because I'm overstaying my welcome here, but I want to talk about some diseases now. If you look at humans, if we have a mutation in a tRNA modifying enzyme or a loss of a tRNA, that deficiency can cause dozens of different human diseases. One of them, most prominent ones, are neurologic diseases, mental health, neurobiology development, very serious human diseases caused by this. What if we overexpress a modifying enzyme at the wrong time? This is now a big deal with cancer. And so colleague Richard Gregory, now at UMass Med School, he's chairing their RNA group there. His postdoc, Esteban Orellana, is now at Dartmouth as a junior faculty member. They published a paper a number of years ago showing that many cancer cells overexpress metal-1. They have gene amplification, like cancer cells often do. 400-fold amplification of metal-1 means you're putting M7G at a position in 17 out of 250 human tRNAs. Only 17. M7G stabilizes tRNA against degradation. The number of copies of that tRNA go way up. They measured them. And so in these cancer cells that overexpress this, they have whopping high levels of those tRNAs. What codons do those tRNAs read? They read codons enriched in oncogenes. Cool. Tumor growth promoters. Well, Tom Bagley then did an analysis of Go categories. Tumor growth suppressors in red, tumor growth promoters, oncogenes in green. They have exactly the opposite codon usage patterns. So this tells you that we're now making too many tRNAs at the wrong time that are reading the codons enriched in growth promoting genes. Richard showed that if you knock out that enzyme or knock out one of those tRNAs, the tumor stops growing because it's not translating growth promoters. So we have a company going to make inhibitors of overexpressed enzymes in cancer to stop the growth. Now we can use radiation, chemotherapy, other things to kill the tumor, or the immune system can come in to do that. But all these different kinds of cancer have subsets with overexpressed enzymes due to gene amplification that we can now make drugs against. We have a pipeline of 20 different targets to start making anti-cancer drugs. Well, microbes, I told you, parasites, even viruses screw around with our systems. They hijack the system. Bacteria. Here's a classic one. A lot of bacteria, especially gram-positives, have erythromycin-resistant methyltransferases that put a single methyl group on ribosomal RNA at the peptide exit tunnel, a methyl group. It prevents macrolides like erythromycin, the WHO's most important antibiotic, prevents them from binding to the ribosome and inhibiting it in the bacteria. So a dumb, single, stupid 14 Dalton methyl group is inhibiting the most important antibiotics in the world. It's an RNA modification. We said, let's go look for things that reverse that and restore sensitivity in resistant microbes to this. Well, it turns out that in Mycobacterium abscessus that causes non-tuberculous mycobacterial infections, which are almost worse than tuberculosis around the world, 50% of them have resistant strains. We've come up with a drug now that reverses restores the activity of erythromycin and clarithromycin, and you can inhale it. We're making an inhaled version to treat this and improve the activity of this antibiotic. Another example would be tRNA modifications that are in humans, but the human enzyme is structurally very different than the bacterial enzyme. Now you have selectivity to make an inhibitor. If you knock that out and it's an essential gene, the bug dies. So we have a program going to make antibiotics against that. Now, I want to point out that a lot of the tools we talked about can be used at all the stages of drug discovery and development. First, we got to have a target. Well, it's TRIM-D. Great. Okay, there's our target. We got to go find molecules that inhibit TRIM-D and get in the bacterium. So let's do whole cell phenotypic screening. Well, once we get a compound that seems to have activity, we have to make it safe in humans and more potent against bacteria, hit to lead. We then test it in animals. All along the way, our tools can be used to say, did M1G go down with our TRIM-D inhibitor? No, it didn't. Go back and redraw the molecule. So we use these all along. We go find our patients for clinical studies in cancer. Which patients with cancer have overexpression of the gene and too much modification? We can use it as a diagnostic tool. So we have a team now at MIT and in Singapore to do drug discovery and development for antimicrobials. We've got a professional medicinal chemist. There's Tom Bagley. Tom and I are like Siamese twins, joined at the hip in that sense of being partners, a professional drug developer from years, people at the Broad Institute for Cancer. Boon Chong Go, a former postdoc, has his own company making deodorant. Of all things. He then patented license on phages that selectively kill odor-causing bacteria. They're very potent, selective antimicrobials that don't harm your microbiome. So you can kill odor-causing bacteria. He's got a company, AeroBiome, that is now winning awards at all the cosmetic shows. It's cosmetics, right? Well, I'm sorry. Go do something practical with your discoveries. Anyway, I digress. I'm going to really quickly jump to the thing. We're doing whole cell phenotypic screening for bacteria and human cells. The point of putting the whole cell in there to screen for compounds is the compound has to get in. So if you get a compound that gets in and gets to its target, you get a signal. So if you screen for compounds that don't get in, you don't get any signal. Bacteria are horrible for getting compounds across gram-negative cell walls. So you got to do whole cell phenotypic screening. And then if you get an activity, bingo. So you solve all kinds of equations by doing this. Same with cancer cells. If I can put cancer cells in there, throw on drugs, I know they get in, I know they get their target, and I know they have activity. So we do a lot of that. Where do we get the compounds to test? We're not going to go screen 1 million compounds. We've come up with now a 10,000 compound library that our medicinal chemist, Yannick Lacroix, collaborated with Enamine, the Ukrainian compound company. Imagine a company in the middle of a war helping us screen their 6 billion compound library and helping us through all kinds of computational tools create a 10,000 compound library of RNA modifying enzyme inhibitors, high probability of being epitranscriptome inhibitors. We just received the library, 10,000 compound library from them a week ago. It's amazing that they're surviving in the middle of a war. It's incredible. Go Ukraine on this one. Big thing. First 130 compounds, we couldn't afford to buy the whole set. We just bought 130. Immediately got 3 hits. This one inhibits cuisine, galactosylated cuisine and cuisine. None of the other modifications are affected. We don't have any toxicity. It's got a therapeutic index of about 10. We hit another one that it goes after metal 1. It reacts a little bit with M2G, a bit, but much more potent with metal 1. It's got an IC50 against cancer cells of 14 micromolar. It's toxic in human cells at 410. So we have a 30-fold selectivity index. We haven't done any medicinal chemistry yet. That's just one of the hits. So we have 3 hits out of 100. That's about a 2% hit rate. Typically from screening big libraries, it's 0.05 to 0.1%. So I think our library is really going to crank out specific inhibitors. Here's a compound that's being tested by a competing company against messenger RNA. It is potent against M6A in messenger RNA, but look at all the peak changes. It's causing significant toxicity. So our system allows us to see toxic effects, and we're going to make an AI model that correlates structure of the compound with cell toxicity. That's it. Thank you very much for listening. all the great people in the lab who've been doing this, all the people in the past here who have gone on. Tom Bagley and Red Highlights, my best friend in the world. And thank you very much for your time. I appreciate it.
Wow,
Thank
I
you.
wanted you to continue.
Yeah, sorry
Thank
to
you.
make it
We're
long
very
here, but
excited.
it's just both. I wanted to hit both worlds for microbes to touch on them.
Oh, that's great. Thank you very much for a very exciting presentation, Professor Dedon. Professor Matty, would you like to?
Yes, yes, I will get this rolling. I did not want you to stop, Professor.
Thank you.
I'm going, keep going, don't stop. And it's
See,
like, yeah.
I have my coffee. I could go all day. I have my cup of coffee, you know.
No, I want you. I'm going to send you an invitation for another conference, and but it was an amazing talk. And you didn't talk about this in Thailand, right? You completely touched upon something else. But what we talked post-conference was that how you have taken this basic mechanism, the tRNA mechanism, the tRNAs, and developed these amazing tools that not only led to so many discoveries, but how it gets translated to diseases, antimicrobial resistance and cancer. I think that's what caught my fascination then, and it is still my fascination with you. And so I really, really appreciate it. But I could not help noticing something.
Okay.
So I'm an Aeruginosa person, right? So I spent 30 plus years working in Pseudomonas aeruginosa and debunking a lot of myths about it. And I saw the two genes, MUC-B and MUC-D, in your collection of genes. Okay? I forgot which slide number. Oh, you didn't have a slide number, but this was on your collection. Okay, let me tell you something. The thing that kills cystic fibrosis patients is not the infection per se, but the infection where the bacteria fights back and produces alginate. Why does it do that? Because our host neutrophils are constantly bombarding against Pseudomonas, now has established presence in the lungs, right? So my question is, how much sulfur is in the lungs? Because these genes are in your list. So I don't know the answer, right? So, and the question is, then I said, when we knock out MUC-B and MUC-D, and my research involved that particular operon, so I know quite a bit about those two genes. When you knock out those two genes, the alginate production goes up. This is the polysaccharide in the lungs, prevents the access of antibiotic access, as well as the host immune response access to remove the bacteria. And CF lungs patients, of course, they don't have a cilia. The cilia doesn't work, so they don't lock off. So sorry, it's a lecture. I apologize.
No.
So I'm going, God damn it, we need to focus on MUC-B. And these are periplasmic proteins, right? Which is even better. You hit at the periplasm with a drug, but then if you knock out these two genes, the alginate production goes up. So now we need to do the opposite. You don't knock the genes out. You try and figure out how to increase their expression in the system. But it is nasty. We can talk about it all day long, and I can happy to talk to you about the whole circuitry. So, do you have any comments? I should stop here.
So sorry, what were the names of the genes?
MUC-B and MUC-D.
Spell that.
MUC-B, MUC-D. It's in the list of your genes where you had the modifications, the 3 groups, and you focus on those 3 groups. And I'm looking at which one of the genes I recognize, right? So that we can talk about it. Yeah.
I'll go back and look to see. Well, they must have been involved in the MIA-B, in that I6A pathway. And we didn't put those into the diagram. I'll go back and ferret out the biochemistry. This is what Jingjing has been doing now with all these pathways that showed up. There's a whole project just to go explore those pathways. How do tRNAs regulate this periplasmic, macrophage-related, resistance compounds? Yeah, I have no idea. And that's, all we've done at this point is to sort of find the pathways that mess with something we can measure. Now we have a metric. And now we can go back and study these pathways and get more into, are there other genes along these pathways that are maybe essential that aren't in our library? And we can start to piece them together and then start to find targets potentially that, for drugs like, or overexpression. How can we increase the expression of these things to restore? The other thing we're doing a lot now is biofilms. I have a postdoc in Singapore who's looking at Enterococcus faecalis biofilms and how the tRNA modifications reprogram as you go from lag phase stationary all the way out to biofilms. And there's some amazing changes. So maybe, and Pseudomonas makes biofilms, right?
Of course.
Nasty ones. So yeah, I'm with you. I wish I wasn't so old and there was more money and students to go get all these pathways dug out.
I know. I feel the same. Don't worry. I feel like I want to go back to the lab and I want to send all my mutants to you and say, go figure this out. And I think it'll open a whole new set of drug targets. It's an amazing. Go, Jose. I know I can talk a lot.
We have a question from the audience, Professor Ram. Have you tried verifying Koch postulates for IBD by introducing mutant bacteria unable to carry out, different types of RNA modification into non-biotic mice?
That's a great question, and it actually predicts exactly what we're thinking we need to do. We're not going to be able to go into humans. We do have to go into mice first. Inflammatory bowel disease patterns, to your point, let's go in with mutants that don't make. Let's replace. We can actually get the same genetic background and simply knock the restriction modification system out and put these into gnotobiotic mice and compare different reactions in genetically engineered inflammatory bowel disease, just in how these bugs affect physiology in the mice in general. How well do they survive in the gut? So yeah, it's a great predictive tool for us to be able to start doing that. Yeah, and I just wish we had a granting system in the U.S. now that that valued this kind of work. So we'll see. Thank you for the question.
Yeah, things will change. We are very hopeful things will change. But I am also fascinated. Sorry, Professor Pente. Fascinated the high hit rate in the Ukrainian library. Was there a specific rationale they used to create the library?
Yes.
Go ahead, please. Yeah.
Oh, yeah. I didn't go through that. So what we did was to go through all the literature, go through all the patents, and pull out all the compounds that were known to inhibit RNA modifying enzymes. So Storm Therapeutics has the structure and many other structures of compounds that inhibit the enzyme that makes M6A in human messenger RNA. So we pulled all those structures out. Those are known RNA modifying enzyme inhibited structures. Then you use machine learning models. You use algorithms that there was Tanimoto similarity index. So you look for chemical structures that kind of look like it. You use the Merco algorithms, all these chemical structural tools to go through and go from 6 billion down to 10,000 that look like and are predicted to behave like RNA modifying enzyme inhibitors. You pull out all the ones that are known to be bad drugs. There's called pains. They're things that react with P450 to cause toxic reactions. Get rid of those. And so you boil the ocean down to get these 10,000. And right now, I know Jingjing is screening the other 9,900 that we didn't buy. And I had hoped to have some data. I didn't push her. I didn't say, hey, where are those 9,000 compounds? But the first 130 have already turned up 3 super potent compounds. So
Yeah.
we're really hopeful.
Yeah, I see that it has a dose-dependent inhibition, and that's amazing. You not just found the compounds and immediately show they are dose-dependent, and also at the levels low enough. Now I think we need to go, how low can you go before, that you can still have the same effect that you see? Because you had a 14 micromolars, right? We
Yeah.
can, we can even go lower with that. And that's amazing because, when you use molecules, very making a conscientious effort to reduce all the pains, etc., then you, of course, get a fantastic hit rate. Um, and I mean, we did a screening using plants that people use it for treatment in an ethnobotanic approach.
Right.
We had a 6% hit rate when
Yeah.
we ethnic an ethnobotanic screening. So this is amazing. I'm just awed how successful, and let's hope Ukraine survives this.
Really. Yeah, it's funny. You mentioned screening plants. Being in Singapore, Chinese traditional medicine is huge. And the
Yes.
universities all have departments studying this, and there's shops all over Singapore. I wanted to do a project with one of my Singapore Chinese heritage Singapore students on taking classes of Chinese traditional medicines with specific properties and put them through a screen now. And let's look at their epitranscriptome reprogramming, see if we can't find interesting things in there because they're very potent medicines. So, like
Yes.
your plant screening rate had a high hit rate. Yeah, sure. Traditional medicines are real.
It is real. I tell you, I only go to Western doctor if I need an X-ray. Otherwise, I go to Chinese.
But we have a couple of additional questions from the
Okay, go
audience.
ahead.
Are these applications available in the clinical setting or just research setting only, especially concerning AMR?
Yeah, we're starting in the research setting and translating now, no pun intended, into the clinical setting. That's the goal. Uh, that drug development scheme starts with in vitro screening against bacteria. We have collaborators with mouse models of different kinds of infection that we use for pulmonary infections, uh, our non-tuberculous mycobacterial infections. We have a model where they do an inhalation infection and get it with Mycobacterium abscessus. The mice are genetically engineered to set up a chronic infection just like a human. So now we have both. So now we can deliver with small little prongs in the nose of the mice. We can deliver powdered antibiotics just like a dry powder inhaler, follow the efficacy and growth of the bacteria killing. So we're translating now into that clinical setting, hoping to come up with something that gets into a real human clinical trial. So we're trying, we're on the way.
Yeah, your work is an excellent example of how teamwork actually can make science go ahead, and the tons of applications and potential useful molecules to use in the future is just amazing. Professor Sitaraman has an additional question, and I have a related question about it. But he says, I'm not clear about the open structures of genes encoding modifying enzymes, but I wonder whether the phenotypes of some of the transposon mutants from the library aren't actually due to some polar effects. Have you cross-verified by making a verifiable non-polar mutant by homologous recombination, for example?
Very good. Good question.
Yeah, not for all of them. We did have early on, and in the paper, I think we discussed quite a few of first of all, the annotations of the genes were wrong in the transposon library. Some of them, that transposon inserts in a weird place, and you do screw up neighboring genes, and that neighboring gene is the one that's causing the tRNA modification, just as you're saying. So we haven't sorted through all of them, but we got quite a few. And then all our positive controls, where the dozen or more tRNA modifying enzymes, we hit them and got appropriate changes. So yeah, there's still some de-weeding, debugging to do for some of these genes. We don't think there are too many where there's going to be, innocent bystander effects. in these
Right.
transposon libraries. And the next thing to do is to go into a different form of library and do selection, where you have all the mutants barcoded and mixed together, and now put them in a mouse and see what comes back out and measure the modification. So, yep, not
So,
perfect,
It's a
but pretty good.
yeah.
regarding the library, you're going to have the antibiotic resistance being expressed from different sites in the chromosome. Do you think the expression, the RNA from the resistance gene might have an influence on what you see with the modifications on other genes?
It's an interesting question. Um, could there be sort of an indirect effect
Yeah.
of just the RNA made from resistance genes as you stimulate them and the resistance gene is activated, like ERMs? ERMs are inducible from that
Right.
standpoint. What modifications around those RNAs in some way does that shift the pool a bit? I don't know.
Yeah.
Yeah.
It's a very interesting comment, question, Dr. Pente, and also from Dr. Sitaram. I'm very familiar with the library, used the PA14 library, the Ossables library, because we used it too. The biggest problem is you need to go back and do a point mutations or in-frame deletion, so that it's really not a polar effect. It's definitely. But I can tell you, MUC-D is the last gene in the operon. So,
There is an interesting feature, though, in our mycobacterial studies, that the genes in the operon have unique codon biases. There's not an operonic codon bias. Each individual gene has a different codon bias. So when you get the
wow.
polycistronic messenger RNA, the other genes are not getting selectively translated. The ones with the right codons are. So there's a really interesting feature of that, and this is in our original paper on Mycobacterium bovis hypoxia. The student had to go through and analyze all the genes that went up. Which operons were they on? What are the codon usage patterns of the other genes in that operon? And now you've got your regulon of different genes, but then the operonic structures, some really interesting codon biases. And how did that happen evolutionarily? That they're in the same operon. I'm not a microbiologist, so this just blows my mind. So
No, it's really a new an insight that I never thought about it, right? Because we always thought the level downstream levels go down because the stability of the RNA is the processing of the RNA molecules is from the 3' end, not at the 5' end. So then the levels are controlled, but then of course it's always the last gene in an operon. Usually is the regulator or the rate limiting gene product for any major operons, very large operons, like Pseudomonas algae operon has 12 genes on it, and the rate limiting gene is the last gene, right? So it's for me, knowing genetics, it makes intuitive sense, but to know that at the RNA level, things are changing. I have a colleague who insists, don't tell me you see this gene go up based on the RNA. How do you know the protein is made? And he is like all about proteins. If you show me the proteins, I'll believe you. Otherwise, I'm not going to listen to you. It's true.
Yeah. We always do proteomics to accompany this to make sure, did it get made?
Exactly. Wow.
Your presentation was so rich on many different aspects of what you're studying, and there are plenty of questions that can come up from it. And unfortunately, we're running out of time. But I would like to go back to what you mentioned at the beginning regarding. You mentioned that actually 5 to 10% of the microbes of the microbiome has these PT modifications. And probably you alluded about it. Do you have any idea of the selective pressures that keep or maintains the PT systems and this kind of intermediate level?
We don't, and that's now the deep dive into this polymicrobial communities. What is it that phosphorothioate containing bacteria are making that other bugs use? And maybe it's hydrogen sulfide. They often, other bugs will pick that up and use it. What things are other bugs making that the phosphorothioate bacteria need? So this interdependence, we haven't figured that out yet. I had a postdoc who was big on the informatics of microbiome and metagenomics and all of that, and he moved on, which is people should, right? They should go get real jobs. But he started to do those kinds of interactome things. We never have gotten to an experimental model where we could like play with, like, feed a high sulfur diet to mice, or a low sulfur diet, and see what do we do to those phosphorothioate bacteria. So yeah, there's a huge amount of work just to figure out.
It also comes down to the question why firmicutes are less in these PT modifications. So what are the restrictions there? I guess are the genes or how they are expressed.
Well, and that's, for example, we haven't. The postdoc who did the nearest neighbor analysis to show the hydrogen sulfide gene associations with PTs, I forget how he fractionated them into bacteroidetes and firmicutes. Maybe the firmicutes just aren't the hydrogen sulfide producers, and that's essential for survival in this IBD population. So many possibilities here. And it's very surprising bias.
Yeah. So I want to just throw something as the last questions, because we are going to run out of time. Is that, my always I think about is that, so I want to go in every disease systems where bacteria plays a role, right? What is the sulfur content? I don't think anyone, at least in a Pseudomonas field, actually looked at how much in the environment, the milieu, there is sulfur, and then correlate it with the disease levels, and then figure out how do we soak up the sulfur so that it prevents this overgrowth of any bacteria without having to destroy all good and bad bacteria, right? And I think that's what I will throw at you. And I'm going to let Dr. Fente wrap up.
Okay, we can go on. Unfortunately, the time is limited. Well, okay, let me close up. Our sincere thanks to Professor Pete Dedon for today's insightful presentation, and to all of you for your thoughtful questions and active participation. A link to the recording will be emailed to all attendees shortly. We hope you will join us for the next session of the JMM seminar series, which will take place on Friday, March 27 at the same time. Dr. Robert Fagan from the University of Sheffield will present his research on the evolution of vancomycin resistance in Clostridium difficile. Thank you, Professor Dedon. It was very exciting, and thanks for being with us. Thanks to everyone. Thanks, Professor Matty, for your enthusiasm. And thanks to Fiona for her assistance. And well, we hope to see you next time.
Yeah, and
thank you very much.
Great start for 2026 series.
Thank you. It's a big honor, and I really appreciate it. Thank you.
Thank you very much.
We appreciate you. Thank you. Thank you, Professor Dedon.